In the last section of our lecture, we're going to talk about alterations in chromosome number and structure. So we're going to be focusing on mistakes that can happen during meiosis and what that does to the number of chromosomes in the gametes that are made, and then if those gametes get fertilized and make an offspring, what kind of problems might the offspring have? So there's a special name for when chromosomes basically stick together, and it's called non-disjunction. So think about disjoining, meaning breaking apart. That's what the chromosomes are supposed to do in mitosis and meiosis. But in non-disjunction, they don't split apart like they're supposed to. Now there's two possibilities of how this could happen. A, the two chromosomes in meiosis 1, which are supposed to go to opposite sides like this, they sort of get stuck together and they both go the same way. So now at the end of meiosis 1, both of these chromosomes are here, and this cell basically didn't get either one. What's supposed to happen is that each of these cells would have one. So we would have started with a cell with 2, and each one would have 1. That would have been normal meiosis. In this case, both of them, they got stuck together and they both went the same direction, and this other cell didn't end up with any. Now that would be if there was just 2 chromosomes. So in us, if you think about it, we have 46 chromosomes, or 23 pairs of chromosomes. So in us, imagine that, our 46 chromosomes line up in 23 pairs and one pair sticks together. Then what's going to happen at the end of meiosis 1, we should have gotten 23 chromosomes and 23 chromosomes, right? 23 went left, 23 went right. But if just one pair sticks together, this one would now have 22. This one would now have 24 chromosomes because 2 of them went one way, and this one would only have 22. This one would be missing a chromosome, the other one would have an extra one. The other thing that could happen is, if it happens in meiosis 2, the chromatids stick together. So you have the chromosomes and they line up down the middle. And remember, in meiosis 2, it's like mitosis. The actual X's, the chromatids, are going to split in half. So what should happen in this cell is you should get 4 and 4. 4 sticks, or chromosomes that are now, just a stick, they don't have chromatids anymore. And 4 in this one. But if 2 chromosomes stuck together, what could happen is you could get 1, 2, 3, and then instead of 4, you actually get a 5th one. So now this one has 5, and this one, it got half of this one, half of this one, half one, 1, 2, 3, but it didn't get either. It was supposed to get 1 from here, and it didn't get 1, so this one only has 3. Either way, this one is missing, and this one's missing, and this one has an extra one. And so what could happen, this says, explain how this could give you a diploid gamete. That would have to happen. The only way that could happen is if the chromosomes lined up and all of them stuck together. So if all of them stuck together, instead of 4 going this way and 4 going this way, if all 8 went one way and 0 went the other way, this gamete would actually still be diploid. It would still have 2 copies of every chromosome. So that would be the only way that could happen. All right, so this picture kind of shows it better, I think, than mine. Let me just erase the ink here. So this is showing non-disjunction during meiosis 1. So what was supposed to happen here, just to clarify, is the chromosomes would have lined up side by side, this pair, this pair. This would have been meiosis 1. And then each new cell would have gotten 1 copy. They would have gotten split in half. And then in meiosis 2, the chromosomes themselves, the chromatids would have split from each other. And so at the end of meiosis, this cell should have had the correct number in for this cell would have been 2. Because the diploid cell, 2n, was 4. So the diploid number was 4, the haploid number is 2. But if in meiosis 1 these two stick together, then what ends up happening is instead of them getting 2 and 2, this one now has 3. It has an extra one because it got 2 copies of this one. So it has n plus 1, the haploid number, but plus an extra. And this one also has that. The other one got the haploid number minus 1. It's missing a copy of that second chromosome. This chromosome here, it doesn't have a copy at all. And this is showing what would happen if it happened during meiosis 2. So in meiosis 2, so they're saying the original chromosome split. This is a cell that normally would have a diploid number of 4. Each of these has 2. Then the chromatids are supposed to split. But if you look at this one, the 2 chromatids, they stuck together, and they both went the same direction. So this cell ends up with an extra chromosome. This one is missing the other chromosome. But the other 2 are normal because this one is going to split normally. So if it happened in meiosis 2, only 2 of the gametes would be abnormal, and the other 2 would be fine. Okay, to look at abnormal chromosome numbers, we use something called a karyotype. Just know what it is. We isolate the chromosomes, they take a sample, they isolate the chromosomes, and then they pair them up, and this is what it looks like. They stain it with a stain, and scientists, we've actually come up with a system of numbering the chromosomes. So pair number 1 is always going to be the same chromosomes that carry the same information every time. So if we know that a genetic disorder is located on a particular chromosome, like cystic fibrosis, the gene is on chromosome 7, that's what they mean when they say chromosome 7. It's the chromosome pair that scientists call 7. They pretty much go from big to small, and then there's also like a pattern of bands that you can't see really well. These are X's. They're just that the 2 sides are they stick to one another, so it doesn't look like sometimes when we draw the X's in class, we draw like that. In reality, the 2 sides sort of stick together, but you can kind of see there's a little gap there. So these are actual X-shaped chromosomes. Now the last pair, the 23rd pair, they didn't number it 23. That's where they put the sex chromosomes. This is a boy. It has a big one and a little one. That's XY. A female karyotype would look a little different. It would have 2 that looked like this. It would be XX. So this is a male, and then XX would be a female. And the next couple slides will show this. Now, a karyotype could tell us, if you got a karyotype of some of these chromosomes, if they had an extra or a missing chromosome. So, for example, a very common disorder would be Down syndrome, and they have an extra chromosome 21 here. And how did they get that? Well, they got that because of non-disjunction. Either the sperm or the egg, one of them, had that problem during meiosis where that parent gave 2 copies of chromosome 21. There should have just been 1 copy from this parent, 1 copy from this parent. But a mistake happened, and this parent gave 2 copies. It could be the mom or the dad. And so then when the sperm and egg get together, now there's 3 copies of chromosome 21, and so that would be Down syndrome. Now there's lots of different syndromes, not just Down syndrome. That's just a very common one, so we use it as a kind of a textbook example. This is just showing the process. You're not going to be tested on this. They use a special solution. I've actually made a karyotype of my own chromosomes before, and they had to take a blood sample for us to do it, and they gave us the culture, and then we did this part. They gave us the solution, the stuff, and then we stained it ourselves. And this is just somebody looking through a microscope, and you can see over here on the computer, they're placing those chromosomes where they go. This is another picture. Again, this is a male again, XY. And the checkpoint question, how would this be different if it was a female? Well, this is a male because it's XY. A female would be XX. So that would be the difference between a male and a female. All right. The most common disorder where there's extra chromosomes is trisomy 21. It's called tri because there's 3 copies of chromosome number 21. And we call this Down syndrome. This is what it looks like in a karyotype. See how there's 3 of those. So this is a girl. She has 2 X's, XX, not XY. And then she has this extra chromosome 21. Most disorders involving an extra chromosome, as far as I know, are fatal. Like an extra chromosome 13, an extra chromosome 9, the mom would probably have a miscarriage and never even give birth. They cause really bad problems, which a lot of people are surprised. You think it's just 1 extra chromosome, but apparently it causes a lot of problems. Down syndrome obviously causes some physical features and some mental impairment and some problems, but people with Down syndrome, they can hold a job, they can go to school. I mean, for the most part, they can live a fairly normal life. So it's one of the disorders that does not have severe complications. This is just showing that there is a relationship between how old the mom is and the chances of having a child with Down syndrome. And notice by the time you get to about 50 years old, the chance goes up to about 80 births per 1,000. So according to this, it would be about 8% of the infants are born with Down syndrome when the mom is 50 years or older. And the reason why is because you have all of your eggs when you're born. It's just that when you go through the monthly cycle, one egg ends up going through meiosis. So every month, you only have one egg go through meiosis and mature, but technically all your eggs are there, and so they're aging with you. So the older you get, the bigger a chance that when the eggs go through meiosis, there'll be a problem. Again, still only 8%. It's not a super high number. Non-disjunction can also happen with the sex chromosomes. So there is such a thing as where somebody gets an extra X or an extra Y. There's even a case where somebody only gets one sex chromosome. So their sex is X0. They don't get a second sex chromosome. XXY is called Klinefelter syndrome. That sometimes causes, there are some side effects to that. There are other cases like XXX or XYY where the offspring pretty much is normal. So we know that sometimes a chromosome disorder in the sex chromosomes may or may not even cause some, a difference. You might not even notice it. And just here are some examples. You're not going to be tested on these, but so XXY, it's called Klinefelter syndrome. You are a male. The presence of a Y chromosome actually does make a person develop male automatically, but they do tend to have some. It says secondary female characteristics. Mainly, what it is that they don't grow body hair very well. They carry fat on their body more like a female. They may have some fat production in the breast tissue, but they don't look like a girl. They're not a transgender person. They're not, pretty much, you could see somebody on the street that had Klinefelter syndrome, and in a lot of cases, you would not recognize that there was anything genetically wrong with them. XYY, often nothing happens. There's not any physical features. Triple X, sometimes no physical features. Sometimes they're taller than normal. And then X0, this is called Turner syndrome. This is a girl. Again, if there's no Y chromosome, you automatically would develop female. They do have some problems with going through puberty, but they have other problems. I had a student with Turner syndrome before, and I know she had some heart issues. She had some other issues. Mentally, she was perfectly normal. She was actually one of my brightest students, but she did have some physical impairments. So, non-disjunction can also cause what's called polyploidy, but that's very rare in animals. They're showing a frog here that's tetraploid. That means it actually has 4 sets of chromosomes. It's much more common in plants. A lot of times when you see giant strawberries, big watermelons, bananas, a lot of the fruits that we eat today come from plants that are polyploid, that actually have 4 sets of chromosomes, 8 sets of chromosomes. But it's much more rare in animals. You can't live with an extra set of chromosomes. There's no humans walking around with 96 chromosomes. They're not like the X-Men or anything. That you just don't survive. And there are also problems, and this really isn't going to be big on your test either, where you don't just have extra or missing chromosomes, but there's a mistake within a chromosome. Like there's a piece of information missing, or an extra section, a copy that got copied twice, a part got flipped over. Part fell off of one chromosome and got attached to a different chromosome. And so reciprocal translocation, this is where a piece of chromosome breaks off and ends up on another chromosome. The reason it's different from crossing over is because crossing over is where a piece of chromosome swaps places with its partner. This disorder would be where chromosome number 6 somehow ends up swapping a piece with, say, chromosome number 19. Those are not matching partners. So these genes don't match up with what's supposed to be there. And so that's not the same thing as crossing over. And these are just some pictures of some of those things. So a deletion, you actually are missing a piece of a chromosome. A duplication, you actually have a section twice. An inversion, a piece of the chromosome gets flipped over. So the DNA is pretty much backwards. And this is the translocation, where a piece of a chromosome ends up on a non-matching chromosome.